Hi all, Tony here from Immortality Cryptocurrency. Today we're at New Atlas. Article reads, Diabetes Breakthrough Restores Insulin Production Using Existing Drug. A new study could help restore insulin production to the pancreas of patients with type 1 diabetes. Australian scientists have demonstrated a new way to restore insulin production in pancreatic cells using a drug that's already approved for use in humans. The study could mark a major breakthrough towards new treatments for diabetes. Bit of speculation there. Blood glucose levels are managed by the hormone insulin, which is produced in beta cells in the pancreas. However, these cells begin to die off in patients with type 1 diabetes, resulting in little or no insulin production and a lifelong requirement for supplementary insulin shots to manage the disease. In a new study, researchers from Monash University have identified a new way to restore insulin production in the pancreas in lab experiments on pancreatic stem cell from donors with type 1 diabetes. The team was able to activate them to begin expressing insulin by exposing them to a drug compound known as GSK126. Pancreatic stem cells were taken from donors with type 1 diabetes. This person had passed away, and the pancreas stem cells were taken, possibly put in a dish, and they were modulated to begin expressing insulin, differentiation into beta cells using the compound GSK126. Intriguingly, these progenitor cells don't normally produce insulin, but the drug let them functionally step into the shoes of the beta cells that had stopped working. In principle, a single course of this kind of drug over a few days could replace the need for regular insulin shots in diabetics, possibly still requiring immune suppression. Or not. The team says that the new potential treatment has a few advantages over other techniques currently in use or under development. Pancreas transplants are effective, but subject to organ donor shortages and other complications like rejection. Other teams have converted skin cells into stem cells and used them to produce new beta cells. And although results have been promising in mice, immune suppressing drugs need to be given to prevent rejection. That's a little bit unclear. The new treatment would work much faster, within a matter of days, and without the need for surgery. But perhaps the biggest advantage is that GSK126 is already approved by the US FDA and elsewhere in the world as a treatment for cancer. Its safety profile is already being assessed in clinical trials, which would reduce hurdles down the road for its use against diabetes. That said, the scientists caution that this is still very early days. These experiments were conducted on cells in culture, not in animals yet, so there's still plenty of work to do. Nevertheless, it remains an intriguing new possible tool. Before you get it to patients, there are many issues to be resolved, said Dr. Keith Al-Hassani, co-led author of the study. Quote, more work is required to define the properties of these cells and establish protocols to isolate and expand them. I would think therapy is pretty far away. However, this represents an important step along the way to devising a lasting treatment that might be applicable for all types of diabetes. The research was published in the journal Signal Transduction and Targeted Therapy. The team describes the work in a video below.
Diabetes, global disease burden, 536.6 million people worldwide, 1.3 million people in Australia. Diabetic complications include increased risk of cardiovascular disease, kidney failure, heart attack, stroke, nerve damage, lower limb amputation, and vision impairment. By the time a child is diagnosed with type 1 diabetes, much of their pancreatic beta cells which produce insulin are destroyed. People with diabetes rely on daily insulin injections to replace what would have been produced by their beta cells. Currently, the only treatment is islet transplantation, and while the organ transplants prove to be of clinical utility, the stark reality of donor organ shortage has limited its widespread use. In a world first, Melbourne scientists from Monash University have discovered a way that a drug may lead to the regeneration of insulin expression. Using cells isolated from the pancreas of a type 1 diabetic donor, the human epigenetics research team led by Professor Sam L. Oster were able to reawaken stem-like cells in the pancreas to re-establish insulin expression. Using a drug approved by the US FDA, the human epigenetics research team could regenerate insulin-producing cells in pancreatic tissue donated by a type 1 diabetes donor. They discovered a highly selective pharmacological inhibitor of the EZH2 methyltransferase protein dramatically influenced insulin expression in diabetic donor cells. In damaged pancreatic islets, histone H3 lysine 27 trimethylation, or H3K27ME3, functions to silence the insulin gene. EZH2 is the protein responsible for writing the H3K27ME3 mark that silences the human insulin gene. The refractory nature of the H3K27ME3 mark condenses chromatin and characterizes gene silencing that influences beta cell plasticity and insulin gene expression. In rejuvenated human islets, isolated cells derived from the pancreas were stimulated with a specific pharmacological inhibitor of EZH2. The drug effectively reduced H3K27ME3 content, thereby influencing beta cell plasticity and promoting insulin gene expression. A key advantage of this method over other beta cell replacement strategies include rapid insulin restoration, requiring several days of drug treatment as opposed to other protocols that can take several months. Also, the protocol is ethically compliant with no requirement to isolate human embryonic stem cells, and thirdly, the protocol to restore insulin expression is non-surgical.
Inhibition of pancreatic EZH2 restores progenitor insulin in type 1D donor. So, no one knows why autoimmune diseases happen. There are certain theories. One theory is that the immune system thinks that a cell is a virus or a bacteria because of a protein that it has identified on a bacteria that may be the same protein that is on the cell. And so the immune system thinks that the cell is a bacteria. Other theories are that many times the immune system is activated and there's no pathogen there, and it identifies incorrectly, certain cells incorrectly. Cells that tend to multiply are targeted. Abstract. Type 1 diabetes is an autoimmune disease that selectively destroys insulin-producing beta cells in the pancreas. An unmet need in diabetes management, current therapies focused on transplantation. While the reprogramming of progenitor cells into functional insulin-producing beta cells has also been proposed, this remains controversial and poorly understood. They mean there are stem cells in the pancreas. They massage those stem cells to produce beta cells in the pancreas and function as a normal pancreas. The stem cells themselves will then multiply and replenish themselves. The immune system, seeing those beta cells again there, will then activate the immune system and destroy those beta cells. I would say in an effort that would require still immunosuppression of particular interleukins and also clearing the T cells from the area, which would provide a much better environment for those new beta cells to survive. However, this is not the case here. Here, an alpha cell is reprogrammed into a beta cell. The two most abundant and prominent endocrine cell types are the beta and the alpha cells. Both are essential for maintenance of blood glucose homeostasis. The beta cell produces insulin to manage blood glucose, while the alpha cell releases glucagon, which elevates blood glucose. So perhaps the immune system doesn't target the alpha cell. The challenge is determining why default transcriptional suppression is refractory to endocrine reactivation. After the death of a 13-year-old girl with established insulin-dependent type 1 diabetes, pancreatic cells were harvested in an effort to restore and understand exocrine competence. The pancreas showed classic silencing of beta cell progenitor genes with barely detectable insulin transcript. GSK126, a highly selective inhibitor of EZH2 methyltransferase activity, influenced H3K27Me2 chromatin content and transcriptional control, resulting in the expression of core beta cell markers and ductal progenitor genes. GSK126 also reinstated insulin gene expression despite absolute B cell destruction. These studies show the refractory nature of chromatin characteristics, exocrine suppression influencing beta cell plasticity. Additionally, regeneration studies are warranted to determine if the approach of this N of 1 study generalizes to a broader type 1 diabetes population. Why is this amazing? This is amazing because they're using an agent to affect gene expression in order to regenerate the pancreas in vivo, or at the very least activate the progenitor cells to produce more beta cells. This method of using a protein to alter gene expression is a huge, complicated field that has many applications. Introduction. Type 1 diabetes is a chronic condition in which the pancreas produces little or no insulin. Even though symptoms usually do not appear before 80% of B cell mass has been destroyed, absolute destruction of these cells leads to the dependence of exogenous insulin administration for survival. Two strategies currently exist that focus on replacing the damaged B cell mass in diabetic patients, involving either whole pancreas or islet transplantation. Although proven to be of clinical utility, these therapies face a real shortage of organ donors together with the associated side effects of immunosuppressive drugs. Consequently, current research has focused on the replacement of the lost B cells, and this remains an unmet need for diabetic patients. I also wonder if this becomes trivial, or if this can become trivial. Can there be enhanced function for the decline in organ function associated with aging to, for example, supplement kidney function, pancreatic function for improved blood sugar control, and more? Experimental evidence on the inherent plasticity of pancreatic cells has fueled interest in the regeneration of B cells. Pancreatic duct cells have been suggested as a source of progenitors for B cell regeneration. The master gene involved in endocrine fate determination is neurogenin 3. Neurogenin 3 is required for the development of all endocrine cells that are associated with the secretion of specific endocrine hormones within the pancreas. Moreover, during pancreas morphogenesis, NGN3 induces the delamination of progenitors from the ductal epithelium through an epithelial to mesenchymal transition process. EMT is a key developmental program by which cells located within the epithelial layer acquire the ability to spread and migrate to a distant site to form new structures mediated by SOX1, SOX11. In addition to NGN3, PDX1, pancreatic and duodenal homeobox1 is a key regulator of pancreas development. Its expression in progenitor cells driving the differentiation of all exocrine and endocrine pancreatic cells, including the genesis of B cells and the maintenance of B cell identity. Furthermore, PDX1 is a glucose-responsive regulator of insulin gene expression, where it plays a critical role in the regulation of insulin gene expression. Thus, NGN3 and PDX1 appear to be ideal candidates for strategies that aim to influence core transcriptional events using chemical inhibitors, thereby enabling pancreatic B-cell regeneration as a potential path towards improved treatments for type 1 diabetes. So here you see that the researchers have mapped the epigenetic process and are trying to activate those processes. The mapping of the entire processes and associated processes, and then using chemicals in an attempt to do something. Here, re-establish beta cells in the pancreas. Further on, regenerations of whole organs, improved function of organs that are declining, and so on. Is this a taste of the future of pharmacology, or is this just a novelty? The objective of this study was to investigate the specific regulation of the H3K27Me3 writing enzyme EZH2 that is considered responsible for default suppression, transcriptional gene silencing, critical to the ductal progenitor cell's developmental program by enabling them to differentiate into functioning insulin-secreting beta cells. The feasibility of this approach has been demonstrated in other contexts. For example, Dal, Pra et al. Showed that demethylation of H3K27 is essential for the induction of direct cardiac reprogramming. An independent group has recently shown similar results, that regression by EZH2-mediated H3K27Me3 assists the activation of cardiac genes during human cardiac reprogramming. We anticipate that our epigenetic approach targeting EZH2 in order to achieve B-cell replenishment and thereby improve glycemic control could then be applied to relevant clinical settings. We recently showed that DNA methylation is part of the epigenetic barrier to B-cell differentiation, which underscores the central role of transcriptional regulation in tissue regulation. When this barrier is removed, endocrine cells are able to differentiate into beta cells. This was a major breakthrough, since a major restriction to repair damaged endocrine tissue in type 1 diabetes is that the process of beta cell neogenesis from ductal cell progenitors is limited after birth. While this was an exciting development, we questioned whether compounds that target corresponding epigenetic pathways in mouse models can be translated to the human context. Despite some of the initial study successes using epigenetic modulators in mice, they may have shown limited to modest translatability and selectivity in humans. This is likely to be related to complex inter- and intra-cell population differences between mouse and human pancreas. So here they're suggesting that methylation could be a cause of aging, and that modulating methylation could be a way to activate and to reprogram. A potential source of beta cells was previously demonstrated with the discovery of alpha cell plasticity and the ability of ductal and alpha cells to convert into insulin-producing cells. From our studies, we selectively established the ductal cell and alpha cell ontogeny of these trans-differentiated beta cells by direct lineage tracing experiments. We showed that NGN3 re-expression is a feature of pancreatic progenitors in the duct, and SOX11 is a hallmark of EMT. Equally important was the finding that the alpha to beta-like cell conversion observed in these models induces the re-expression of NGN3 in ductal cells and their differentiation into endocrine cells by reawakening epithelial mesenchymal transition. Moreover, we showed recently that during directed beta cell reprogramming, NGN3 and SOX11 are demethylated. This finding represents a paradigm shift in our understanding of insulin biology and provides new opportunity for future treatment. To our knowledge, there are no published studies that have reported successful induction of exocrine progenitor genes using human pancreatic tissue. The rare availability of fresh human pancreas from 2 non-diabetic adults and a child with type 1 diabetes prompted us to focus on default exocrine suppression, influencing beta cell plasticity. So, what Yamanaka factors tries to do on a grand level, this is broadly the same thing, but it's specific to a goal. It's epigenetic reprogramming specific to getting a cell to become a beta cell in the pancreas, or even activating progenitor cells to make new beta cells. In effect, restoring the function of the pancreas, ideally at some stage, a masterful reprogramming and manipulation of these chemicals to restore or regenerate the pancreas and keep the pancreas functioning at a prime level. Discussion. Taken together, we report that the action of the EZH2 inhibitor may facilitate the development of epigenetic compounds for beta cell regeneration. Not only are the data of direct clinical relevance, but our studies are also important to understand new mechanism of gene regulation by pharmacological EZH2 inhibition. There are significant limitations in the present study. First, the case is an isolated one of a type 1 diabetic child with hallmark insulin damage and significant destruction of beta cells. Thus, it is unknown if the results will generalize. Nevertheless, it was possible to increase the expression of core beta cell determinants, including insulin transcription in exocrine cells derived from 2 adult non-diabetic donors. Secondly, and equally important, it is unclear whether classic silencing of progenitor genes can be restored in long-standing diabetes. The ability to inhibit EZH2 and influence exocrine B cell progenitor transcription to near non-diabetic mRNA levels suggests this is highly likely. In view of this evidence, this study is likely to influence research on using exocrine cells from the pancreas as a potential source to generate de novo insulin-producing beta cells and a new working hypothesis on beta cell plasticity. Furthermore, Due consideration to the potential pharmacological interactions and unforeseen synergistic benefits warrant further studies, which we predict will be an important area of studies pertinent to exocrine regeneration and epigenetic research. While GSK126 is not licensed for diabetic milieu, the primary findings add to previous clinical reports demonstrating superior transcriptional reactivation by restoring histone content. Where the GSK126 treatment for exocrine cells restores the content of other histone modifications such as acetylation associated with the human insulin chromatin domain is unclear. Pharmacological EZH2 inhibition reduced trimethylation but failed to alter acetylation content of K27 histone H3. The opportunity to investigate fresh exocrine tissue from a T1D donor and the availability of a pharmacological inhibitor of H3K227ME3 has allowed better characterization of the refractory nature of exocrine B-cell gene expression. Until now, the process of default suppression in the human pancreas was previously considered but challenging to study. The opportunity to investigate fresh tissue from donors once diagnosed with childhood diabetes and the availability of a pharmacological compound that influences exocrine transcription, presumably by chromatization of the insulin gene cluster, have allowed better characterization of this still obscure aspect of exocrine competence. The stark reality of donor organ shortage limits widespread use of cadaveric pancreatic tissue in clinical epigenetics. This is complicated by the challenging nature of sourcing fresh tissue. While the results we have shown highlight the importance of human ex vivo research, we are also aware of the limitations in the study, where the default transcriptional suppression reflected the epigenetic nature of genes in the pancreas and B-cell precursors needs to be confirmed in a larger series of studies. Previous clinical findings and those reported here show that exocrine, endocrine PDX1 and endocrine NGN3 cells have the capacity to become insulin-producing cells. Because exocrine PDX1, NGN3, SOX9, SOX11 and beta cell-specific markers NDX6.1, MAFA and INS genes expression are restored ex vivo, we can predict that these changes are influencing B-cell neogenesis and that GSK126 targets refractory EZH2-mediated suppression. MAFA is expressed exclusively in B cells and regulates the expression of genes involved in insulin synthesis and secretion. NKX6.1 is a lineage allocation factor that derives progenitors towards a B cell fate, where its expression is restricted to B cells found to be required for proper insulin secretion. Importantly, we established proof of concept exocrine barrier accounts for the inability to restore core progenitor genes that influence ex vivo plasticity despite total B cell destruction. The insulin gene is part of a large chromatin domain associated with histone modifications that function to regulate gene expression in human islets. The ability of specialized nucleosomal structures of H3K27ME3 chromatin implicates the refractory capacity of the INS domain to suppress transcription more effectively than conventional or open chromatin or exocrine cells not treated with GSK126. Indeed, H3K7ME3 content was shown to influence chromatin domain structures involving looping conformation to regulate IGF2 gene expression, although other factors involved in regulating insulin expression in type 1 diabetes pancreata will need to be considered, including incomplete processing proinsulin and data emphasis refractory H3K27ME3 content associated with exocrine competency and default transcriptional expression. The present report has interesting clinical implication for regenerative medicine within the context of type 1 diabetes, since it describes the first example of restoring transcription of the insulin gene, providing distinguishable evidence for beta cell regeneration and possible neogenesis. Furthermore, this work reveals that human pancreatic progenitor cells retain their capacity to differentiate into neo-beta cells from the exocrine compartment that were derived from an insulin-devoid type 1 diabetes donor. These findings show that pharmacological alteration of cell fate decisions through the epigenetic induction of pancreatic progenitor genes hold promise for the treatment of type 1 diabetes by promoting ex vivo beta cell regeneration through assisted epigenetic lineage reprogramming. Inhibition of pancreatic EZH2 restores progenitor insulin in type 1 diabetes donor. So this is it, guys. I mean, here we have the possibility of a new age of drug that functions within the manipulation of the epigenome to provide some therapeutic benefit. It's a taste. It's a small taste, but think of the possibilities. Think of possibilities of a machine, a research machine, or an industry that is moving quickly to produce pharmaceuticals, doing a whole range of epigenetic alteration reprogramming. Not huge ones like systemic Yamanaka factors trying to reverse aging. A whole lot of little things like improved organ function here, improving this there, improving cardiac function. So a very interesting article from New Atlas. Diabetes breakthrough restores insulin production using existing drug. Epigenetic manipulation, looking at the epigenetic process. RNA is involved, the genes involved. Okay, over and out.